Hello everyone, we are from Group 4. Today we'll be diving into a fascinating and emerging field in forensic biology, which is forensic microbiology. Salam and hi, my name is Anna Istiqamah binti Mahmud Resti. My metric number is 2023388131. Today I will share with you some introduction about forensic microbiology. Back to basic, what does it really mean by forensic microbiology? So, forensic microbiology is the study of microorganisms to aid in legal investigations. It is not just about bacteria. In fact, viruses, fungi, and even parasites can all serve as crucial evidence. These microbial traces can help answering five W forensic questions like who was involved, what happened, when it occurred, when it took place, and sometimes even why. Why did the crime happen? And this field plays a key role not only in criminal justice but also in public health investigations, biocrimes, and contamination cases. I am pretty sure many of you have heard about forensic biology, but how does it really started? So the origins of forensic microbiology go back to classical microbiology and forensic science, but it began to stand out in the 1990s. One key breakthrough was the use of PCR or polymerase chain reaction, which allowed scientists to detect and analyze microbial DNA. Later, the development of next generation sequencing or NGS allowed experts to study complex microbial communities in much greater details. Thanks to these advances, this field now supports a wide range of investigations, from identifying pathogens in hospital outbreaks to solving um, bioterrorism cases and even analyzing ancient diseases and historical figures. Last but not least, from me, traditional forensic methods like examining corpse signs or using insect activity, unlike those activities, often lack precision and can be influenced by environmental factors. Microbial evidence, on the other hand, is much more specific and consistent. It helps forensic experts determine the time and cause of death, COD, more accurately, and it can even link individuals to certain locations or objects based on their unique microbiome. So, in short, microbiome act like silent witnesses, helping us find answers that other evidence might miss. That's all from. Thank you.
Hi and Assalamualaikum. My name is Putri Nurul Aisyah binti Amiruddin and my student ID is 2023395755. I will continue the presentation by explaining some of the key applications of forensic microbiology and how microbial evidence is used in criminal investigations. First, one of the most important applications is postmortem interval estimation or PMI. When a person dies, the body undergoes decomposition, and this process is accompanied by changes in the microbial communities present on the skin, in the gut, and in other tissues. These microbes follow a predictable pattern of succession. By sampling and sequencing these microbes, forensic scientists can establish a timeline that estimates how long it has been since death. This microbial clock is useful in cases where traditional methods like assessing body temperature or insect activity are unreliable, especially in buried bodies or in extreme environmental conditions. In addition, forensic microbiology helps in determining the cause of death. According to a study by Su and colleagues, in drowning cases, aquatic microbes such as algae or waterborne bacteria can enter the lungs and bloodstream, leaving behind a microbial signature that supports drowning as the cause of death. Similarly, detecting unusual bacteria or microbial toxins may suggest poisoning or fatal infections. In some cases, microbial analysis can reveal infections that were previously undiagnosed but contributed to death. This application is particularly valuable when external signs of trauma or disease are absent or ambiguous. Next, moving on to individual identification. Every person carries a unique microbiome, a complex community of microbes that live on the skin, in the mouth, and throughout the body. These microbial communities are influenced by factors such as genetics, environment, diet, and lifestyle. According to a study by Yuan and colleagues, when blood or tissue evidence is unavailable, microbial traces found on skin, saliva, hair, or personal items can still provide clues. Microbial DNA recovered from objects such as mobile phones or clothing can be compared with reference samples to help confirm an individual's identity. This technique complements traditional DNA profiling and is particularly valuable when human DNA is degraded or insufficient. Finally, forensic microbiology can be used in crime scene linking. Microbial present in soil, dust, or surfaces vary widely depending on geographic location, climate, and ecological conditions. For example, the microbial profile of soil in a forested area will be very different from that in an urban environment. By comparing microbes found on a suspect's belongings, footwear, or vehicle with that of a specific crime scene, investigators can potentially link or exclude individuals from specific places. This emerging method can support or challenge testimonies by linking suspects to crime scenes, adding a new layer of evidence beyond fingerprints or DNA.
Salam sejahtera, my name is Joanson Wilson, and in this video, I am going to explain about the methodologies and techniques used. The first one is the postmortem interval PMI estimation. In forensic investigations, estimating the postmortem interval or PMI is crucial. In this study, researchers use pig carcasses as they decompose similarly to human bodies. The primary technique for PMI estimation is the 16S rDNA sequencing, which analyzes bacterial DNA. Different bacteria, like proteobacteria and firmicutes, thrive at different stages of decomposition. By sequencing the 16S gene from decomposing bodies and surrounding environments, they can track these bacterial changes to estimate how long the body has been deceased. This method is highly accurate, non-invasive, and valuable in forensic work. The second one is the cause of death identification. Postmortem microbiology, PMM, is essential tool for identifying the cause of death. Techniques like next-generation sequencing, NGS, which is also a 16S rDNA sequencing, blood cultures, and histological analysis help detect infections caused by bacteria or viruses such as E. coli, Pseudomonas, or adenovirus. NGS can also uncover hidden infections, especially in sudden or hospital-related deaths. PMM helps identify drowning by analyzing aquatic bacteria or diatoms, and it can also detect postmortem changes, revealing signs of drug use or disease. And the third one is the individual identification and crime scene linking. Microbial analysis is increasingly used for individual identification, especially when DNA is degraded or insufficient. Scientists analyze unique microbial fingerprints from body parts like skin, hair, saliva, blood, and sexual fluids using NGS. These microbial profiles can link individuals to objects or crime scenes. For example, microbes on touch objects like phone or door handles or on saliva found on cigarette butts can help connect a person to the crime scene. Similarly, microbial communities in bodily fluids at crime scenes such as vaginal or semen microbiota in sexual assault cases can help identify suspects or victims.
Assalamualaikum warahmatullahi wabarakatuh. My name is Azmira Shohada binti Muhammad Saini, and my ID number is 202337577. So I'm going to presenting about the conclusion, which is including the forensic capability of challenges, innovations, and the future prospects. So in conclusions, while forensic microbiology offers exciting new possibilities for human identification and criminal investigation, just like my friends have stated in this previous slides, there are still a few challenges that still need to be addressed, such as limited microbial databases and issues with some accurate identification. This is much because of this some microorganisms that are still not being fully characterized, which makes it difficult to identify them accurately. On top of that, the current reference databases may not include the full diversity of microbial strains that exist in different environments of populations. These limitations of this forensic microbiology can lead to the misidentification or non-measurable when analyzing microbial evidence. At the same time, innovations like next-generation sequencing are pushing the field forward by making microbial analysis faster and more detailed than ever before. Next-generation sequencing, or NGS in short form, is one of the major innovations in this field. It offers much higher data output, better efficiency, and more detailed analysis compared to older methods. This improvement is largely thanks to the nanotechnology, which allows million DNA molecules to be sequenced in parallel. As a result, NGS makes it easier to study a complex microbial communities, even from a very small or degraded sample. Therefore, looking ahead, promising areas like the use of gram-positive bacteria as stable forensic markers shows that microbial forensics has strong potential to become a valuable tool in the justice system. This is because the studies have shown that these bacteria are more resistant to environmental factors than the gram-negative ones. This means that they can survive longer on surfaces of human remains, making them reliable biomarkers for tools like postmortem interval estimation or body site identification, so they may play a key role in developing long-lasting forensic evidence. So to conclude, the following references were consulted throughout our research to ensure accuracy and credibility. That concludes our presentation. On behalf of my group, thank you so much.